Small mammal metabolic protocols in avian medicine are often lethal. Applying mammalian assumptions about nutrition to a bird frequently causes a catastrophic metabolic collapse. This discrepancy exists despite shared neural hardware. Like mammals, avian food intake is governed by a highly conserved integration hub, the hypothalamus. This central processor receives continuous systemic telemetry from across the body. Real-time status updates on energy reserves flow in from the crop, the duodenum, the adipose tissue, and the pineal gland. The hypothalamus translates this telemetry into a binary biological language, orexigenic signals that drive hunger, and anorexigenic signals that induce satiety. The danger for clinicians is assuming these color-coded signals behave identically across vertebrate classes. They do not. To understand avian medicine, we must discard mammalian models and learn the specific physiological language of the bird from the inside out. Locating the avian appetite processor requires moving deep into the diencephalon. This is a map of the primary hypothalamic nuclei. In birds, the functional equivalent of the mammalian arcuate nucleus is the infundibular nucleus, or IN. The infundibular nucleus acts as a master biological scale. It constantly weighs competing systemic inputs before issuing a final behavioral command. One side of this scale contains NPY and AGRP neurons. When these fire, they drive the orexigenic pathway, forcing the bird to seek food. The competing cluster contains POMC and CART neurons. These drive the anorexigenic pathway, suppressing intake to maintain homeostasis. All external stimuli, from mechanical gut strain to hormonal shifts, must ultimately manipulate this cellular scale to alter feeding behavior. In mammals, hunger is driven by a redundant network of overlapping peptides, such as MCH and orexins A and B. The avian model lacks functional responses to those secondary peptides. Instead, it relies on neuropeptide Y as its single overriding driver of appetite. NPY is the most potent appetite stimulant in birds, an absolute biological imperative that overrides other systems. An NPY release triggers immediate systemic changes. It accelerates gut motility to clear space for new calories, while simultaneously slowing the overall metabolic rate. Because NPY is uniquely dominant, controlling AV metabolism requires understanding exactly which peripheral signals have the power to suppress it. Short-term meal regulation begins with the mechanical reality of the avian digestive tract. The crop functions as an early warning volume sensor. As it fills, mechanical strain on the outer wall triggers localized stretch receptors, halting intake before digestion begins. This neural feedback travels up the vagus nerve, striking the infundibular nucleus and immediately dimming the NPY hunger drive. A second chemical phase of satiety begins as food moves into the duodenum. Specialized eye cells in the duodenum release cholecystokinin, or CCK. This peptide binds to the vagal afferent pathways, reinforcing the initial mechanical satiety signal. Simultaneously, pancreatic glucagon enters the portal vein to deliver chemical satiety signals to the central processor. Avian satiety relies on this rapid 1-2 punch, mechanical stretch followed by gut peptides to slam the brakes on NPY. While the gut mechanics appear standard, the endocrine signals for long-term energy balance are entirely inverted in avians. In the mammalian model, fasting triggers stomach ghrelin to spike appetite. When ghrelin reaches the avian brain, it triggers corticotropin-releasing factor. This cascade forcefully suppresses NPY, meaning ghrelin actually creates an anorexigenic effect in birds. The discrepancy is even sharper in the mammalian lipostatic theory, where expanding adipose tissue releases leptin to signal fat saturation. Genomic sequencing reveals that endogenous leptin is not highly expressed in avian adipose tissue. The signal effectively doesn't exist. Consequently, birds regulate their energy balance without a functional leptin to adipose feedback loop. Without leptin to inherently cap fat stores, birds have no internal chemical ceiling for weight gain, leaving them entirely dependent on external overrides to regulate their mass. In the absence of a leptin baseline, long-term avian appetite is dictated directly by environmental photoperiods. Light photons penetrate the skull, bypassing the eyes to activate the pineal gland and deep brain photoreceptors. Short winter days maintain a low activity baseline. As days lengthen, this light-triggered system flares up, activating the metabolic and reproductive axes. Ahead of migration or breeding, the bird system initiates a total neurochemical override using corticosterone. While stress hormones suppress mammalian hunger, this flood of corticosterone massively amplifies avian NPY, forcing extreme hyperphagia. Here, the brain is deaf to satiety signals. CCK and crop distension are ignored as peripheral fat depots expand without limit. Avian hyperphagia functions as a hardwired survival mechanism, a precision-engineered biological strategy for massive seasonal fat loading. The avian cycle then transitions to the opposite extreme, the profound fasting required for incubation. Prolactin surges suppress the infundibular nucleus, creating a deep satiety state. This allows a bird to sit on a nest for weeks without eating, ignoring its rapidly depleting fat stores. This chart shows the predictable annual journey, from the hyperphagic peaks of migration prep to the deep anorexigenic troughs of incubation. Captive avian obesity is the inevitable result of placing a hardwired seasonal hyperphagia engine into an environment with unlimited food and no seasonal variation. Successful avian medicine requires managing the bird's appetite as a rigid seasonal calendar, moving beyond the mammalian concept of a daily thermostat.